So, good evening, everyone. This is Kanika Gupta, your TA for this session, and this is our summary class. Our last class will be tomorrow for week 12, and this is just a recap of what you guys learned in the course. So, we'll just go through it once, what all topics we touched in the course, and what were the different things that we came across, what new things we learned about molecular biology. ठीक है? So let's move ahead with the class. So yes, in the starting week we learnt about cells. What are cells? What type of cells are there? What are the different organelles present inside the cell? How these different organelles जो help करते हैं a cell to work properly, and all of the stuff we learnt about cells in the first 2 weeks. So cell is a higher in a higher eukaryotes has multiple organs to perform specific functions such as liver, kidney, and heart. Organs have specific tissues, and each tissue is composed of cells. So cell is basically the basic unit of life. Cell is the structural and functional unit of life, and it contains all the necessary infrastructures to perform all the functions based on the cellular structure. Cells are classified as prokaryotic and eukaryotic cells. In most of the cases, prokaryotes are single cells, whereas the eukaryotes are either single cells or part of the multicellular tissue system. So cells can be a part of the multicellular tissue system. They can be self-dependent. The whole cell can be a single organism depending upon the complexity of the organism and if it is prokaryotic or eukaryotic. Okay, just a moment. Someone just entered the class. I'll just let them in. Yeah. So this is what a cell is. Next, what we learned in the week first was the difference between prokaryotic and eukaryotic cells. So, prokaryotic cells are smaller in size, while the eukaryotic cells can be as large as 40 micrometers in diameter. जेनेटिक मटेरियल जो जनरली प्रोकैरियोट्स में प्रेजेंट होता है दैट इज सर्कुलर डीएनए प्रेजेंट इन साइटोसॉल एज फ्री मटेरियल while DNA in the form of linear properly arranged chromosomes is present in case of the eukaryotes, which is present in a well defined double membranous structure, that is a nucleus. There is no direct connection with the cytosol of the nucleus has its own ground substance, that is nucleoplasm. Okay. Replication in case of prokaryotes is a single origin of replication, while in case of eukaryotes it is multiple origin of replications are there. Uh, okay, the people who have joined now in the class. So basically, we are just going through of the summary of the course that you guys must have attended the class for the molecular biology course. So this is a summary class. The final class will be tomorrow for the week 12. Today is just the summary class of what you guys have learned in the whole 1 to 11 weeks, and obviously you guys must have seen the videos for 12th week also. So yes, this is a summary class. Okay. Then we learnt about the prokaryotic cell, particularly that is a prokaryotic cell is a much simpler and smaller cell than eukaryotic cells. Eukaryotic cells are more complex. Okay, it lacks membrane bound organelles including nucleus. So it lacks those membrane bound organelles. Okay. Next is just a moment. Yes. Next is the structure of eukaryotic cell. So what do we have in the eukaryotic cell is cell wall is mostly absent in case in eukaryotic cell. Basically they are again of two types. They can be either plant cell or animal cell. So they are much more complex cells and contains many membrane bound organelles to perform specific functions. It contains a nucleus isolated from cytosols and enclosed in a well defined plasma membrane. Okay, so difference between an animal cell and a plant cell is plant cell just a moment, I guess. just message me. Exam pattern, Shiv Kumar. I actually don't know what will be the exam pattern because there's nothing regarding that has been disclosed to us. So I'm very sorry I can't help you on that. If you have any problem in the course in some kind of a topic, then I can help you on that, but not an exam pattern. Sorry. Okay, so if we'll go by the structure, eukaryotic cells, as I told you, are much more complex than the prokaryotic cells, and they are again divided into two types: the animal cells and the plant cell. Plant cells have cell wall, while in animal cells it is mostly absent. The size of plant cell is larger compared to the animal cells. The chlorophyll is present in case of plant cell while it is absent in animal cell. One exception here is euglena. Next is the plant cell have large central vacuoles while the animal cell has smaller and many. Number जो है वो बढ़ जाता है in case of animal cells, while in plant cell it is only single large central vacuole. Okay. Mitochondria, plant cell has few, animal cells has more mitochondria. Lysosomes in case of plant cells are almost absent, while in animal cells they are present. And then these are the further differences. Cytokinesis, like it happens in animal cells by constriction, while in case of plant cell it happens by the formation of a cell plate. Okay, so this is how the cytokinesis happens in case of the plant cell. Next thing that we read in the course is the cellular metabolism. So cellular metabolism is further divided into two parts: the catabolism and the anabolism. So metabolism involves all of the biological reactions that are happening inside a cell, is what metabolism is, while in catabolism it is basically the energy producing reaction, while the anabolism is the biosynthetic pathway. Okay, so it is the biosynthetic pathway for what? So it is the biosynthetic, it includes all the biosynthetic pathway, either it is for carbohydrates, for lipids, for proteins, any of the thing. It includes all of those pathways under the field anabolism. Okay, while in case of the energy producing reactions, these things are broken down into the simpler components so that they can release the energy, and that energy can be used to perform the different functions inside the body. Okay. Next is glycolysis. We learnt about what is glycolysis. Okay. So glycolysis is central to carbohydrate metabolism, and it is the universal pathway found in prokaryotic or eukaryotic cells. It is a breakdown of six-membered glucose into two three-membered carbon to feed the Krebs cycle. Okay. So glycolysis is basically the breakdown of glucose into the simpler compounds into two three-membered carbon sugar. So glucose is a six-membered carbon sugar, and when it is broken down into the three-membered carbon sugar, then it is fed to Krebs cycle for further processing, where the energy production continues. Okay, so in the Krebs cycle is again two modes, either in the presence of oxygen, or they can go for anaerobic oxidation in the absence of oxygen. If oxygen is there, Krebs cycle will run. If no oxygen is there, then these three-membered carbon sugars will go for anaerobic oxidation. So it plays a crucial role for adaptation of a living organism under different types of stress conditions. Okay, it is a 10-step chemical reaction. Then what is a Krebs cycle? Krebs cycle, as discovered by Professor Hans Krebs, and as it has all sugar intermediates with three carbon. So it is also known as tricarboxylic acid or citric acid cycle. Okay, these are the other names that are used for Krebs cycle. In higher eukaryotes, Krebs cycle operates inside the mitochondrial stroma with the different enzymes. In the presence of oxygen, pyruvate formed during glycolysis enters into the Krebs cycle for further oxidation to produce energy, जो हमने अभी पीछे पढ़ा. ठीक है. So this pyruvate must cross the mitochondrial membrane to reach to enter the Krebs cycle. ठीक है. Next is the beta oxidation. Also, we learnt about the beta oxidation is basically sequential removal of two carbon fragments from the carboxyl ends of fatty acid. So, in a fatty acid, there is a carboxyl end. Basically, where the carboxylic acid group is attached, at that end, the beta oxidation happens, and in every reaction a two carbon fragment will be removed from a long fatty acid chain. So that is how the beta oxidation happens, and during the process acetyl coenzyme A is formed as a bond between alpha and beta carbon atoms are broken down. Okay, so it is named so because the beta carbon of fatty acids is oxidized and the process occurs in mitochondria. ठीक है, so this process also occurs in mitochondria. ठीक है, next is the biosynthesis of amino acids. So principally all amino acids are derived from either glycolysis or citric acid cycle or pentose phosphate pathway intermediates. So basically it is the intermediates in all of these three reactions that are the precursors for all of the amino acids that are formed inside the body, which are non-essential. Essential ones are those which we have to take from outside because they can't be formed inside the body. But non-essential are the ones which we can form inside our body from the many precursors that are available from all of these reactions: the glycolysis, citric acid cycle or Krebs cycle, and pentose phosphate pathway. Okay, so these derivatives provide the carbon skeleton for the amino acid, whereas amino group or nitrogen for the same is provided either by the glutamine or glutamate. So nitrogen part comes from either glutamine or glutamate. ठीक है, so depending upon what can be formed inside the body and what cannot be, they are further divided into essential or non-essential amino acids. Those that can't be synthesized in the body are called as the essential amino acids. Okay, next we learnt about the cell cycle. What is cell cycle, how it runs, what are the phases, we all learnt about that also in week 3. Sorry. तो सेल साइकिल में क्या क्या होता है? द यूक्रियोटिक सेल अंडरगोस प्रिसाइज्ड सेल साइकिल एंड डिवीजन to produce two daughter cells. Cell cycle is the series of tightly regulated events leading to the division and duplication. It is a vital process used to single celled fertilized egg is developed into a full organism. So basically this is the process by which a single celled zygote can form a whole new organism. So this process is a very important step. Cell division is the crucial event underlying the regeneration and repair in the tissue, liver and heart. In prokaryotic cells, parent cell is divided simply by division into two halves through the process of binary fission. This process of cell cycle is generally seen in case of the eukaryotic cell. In prokaryotes, it is the binary fission by which cells divide. In eukaryotic cell, cell cycle has three phases: interphase, mitotic phase, and cytokinesis. Okay, so these are the three phases of uh, cell cycle. These phases are further divided into other phases, specifically the mitotic phase, which is further divided into other phases for depending upon the different stages of the cell division. Okay, next we learned about the nucleic acid. What are nucleic acids and how they are formed? So most of the organisms, either prokaryotic or eukaryotic, has a DNA as a genetic material inside them. ठीक है, genetic material means that material which contains all of the information about an organism. ठीक है, so it has the genetic information, whereas a minor fraction, for example virus etc., has RNA as the genetic material. So mostly it is DNA which is the genetic material, but in case of virus etc., smaller organism, a very minor fraction, they can have RNA as a genetic material too. ठीक है. So DNA or RNA is the biopolymer and is acidic in nature. Biopolymer means a biological polymer, ठीक है, which is formed in the living beings. ठीक. So DNA or RNA is the biopolymer and is acidic in nature. In eukaryotic cells, animal or plant, nucleic acid is present within the nucleus, whereas in prokaryotic cell it is present as free form in the cytosol, that we also read in the differences when we were recapitulating the differences between prokaryotic and eukaryotic cells. Okay. So the first nucleic acid that was isolated by Frederick Mischer in 1868. ठीक. Now, what is the composition of nucleic acids? It is uh, one nucleic acid. All of the nucleic acids are formed by a similar monomer, which is formed of a phosphoric acid, a base, nitrogenous base, and a sugar molecule. So this is the monomer of all of the nucleic acids, and that is further then polymerizes to form the whole of the nucleic acid. Now, the phosphoric acid provides the backbone to the polymer, whereas sugar works as an anchoring point for the nitrogenous bases, which actually contains the information. Okay, so the 9-membered nitrogenous bases gives the diversity in the sequence of the nucleic acids. Phosphate backbone or phosphoric acid serves as backbone of the molecule. Sorry, the sugar, the five member here, the sugar molecule is a 5-membered ring, the ribose sugar. Now, the sugar when is present in RNA, it is a ribose sugar. When present in DNA, it is the deoxyribose sugar. So at the 2' position of the sugar, and O is missing, oxygen is missing, so it is a reduced sugar. Okay, so the 5-membered cycling reducing sugar is present in nucleic acid. There are two different variants. The sugar molecule which contains hydroxyl group at 3' are known as the ribose, otherwise, sorry, 3' location, not 3', it is 2', it is written wrong here. It should be 2' only. Okay, so it is known as the deoxyribose sugar. Based on the sugar, nucleic acid is classified either as RNA or DNA. Ribose sugar is present in RNA, whereas deoxyribose is present in DNA. The purpose of sugar in the nucleic acid is to provide the attachment site for nitrogenous bases. Okay, so this is the purpose of sugar in nucleic acid, is to provide the attachment site for nitrogenous bases. ठीक है. Nucleic acid structure. So DNA is a double stranded, whereas RNA is single stranded. In most of the cases, the individual monomer responsible for making DNA or RNA is nucleotide, and as a result DNA or RNA can be considered as polynucleotide because it is a polymer of nucleotides. Individual nucleotide is a nucleoside attached to one or more phosphate groups and can be termed as one phosphate group. Monophosphate nucleoside. If two phosphate groups are there, diphosphate nucleoside, and if three phosphates are there, triphosphate nucleoside. So each nucleoside is composed of a nitrogenous base, a sugar, and they are attached by what? Glycosidic bonds. Tick. So this was information about the nucleic acid. Now, the structure of nucleic acid, if we'll go into detail, so DNA is a double-stranded molecule, whereas RNA is single-stranded. Both strands of DNA are held together by what? Hydrogen bonding, and that happens by complementarity, because adenine will bind to thymine and guanine will only bind to cytosine. This is the rule. Okay, now the sequencing part of DNA. How basically is the sequencing done? So for that, one of the most popular method is the Sanger method. In this, he used actually dideoxy chain terminators, which when added to the different tubes, then different test tubes, and then the amplified templates when ran on the gel, then it was found out that the sequence of this particular DNA is this, because if the deoxynucleotide enters at in the place of the deoxynucleotide, then they won't be able to form the phosphodiester uh, phosphodiester bond with the next nucleotide, so the chain will terminate there and then only. So that is how the Sanger method works. Next, we learnt about the protein structures in this lecture. So, the ordered folding of polypeptide chain give rise to the 3D conformation known as the secondary structure of the protein, such as helices, sheaths, and loops. Arrangement of the secondary structure give rise to the tertiary structure, alpha helix and beta sheets, which are connected via unstructured loops to arrange themselves in the protein structure, and it allows the secondary structure to change their direction. Then the tertiary structure, it defines the function of a protein, enzymatic activity or a nature of the structural protein. Okay, different polypeptide chains are arranged to give the quaternary structure. So primary structure is basically the arrangement of amino acid. Secondary structure is the amino acids when folded to give the 2D structure. Then the tertiary structure is different chains for the same protein when come to interact, it forms the tertiary structure. And then the different sorry the tertiary structure defines the function of the protein, and it is the quaternary structure in which different subunits come to get arranged to form the fully functional protein. Okay. Now, if we'll go into the details of the proteins, enzyme is also a protein, and there are some peculiar properties of the enzyme which should be present in an enzyme to be called as an enzyme. Okay, so it is made up of a protein except ribozymes. Due to the presence of amino acid, it provides specific environment for catalyzing reactions with different types of substrate. The substrate binds to a small pocket within the enzyme. This pocket is known as active site. The molecule produced by the reaction is called as the product, and the enzyme catalyzed reactions are very rapid than the uncatalyzed reaction was. Because what does an enzyme do? It lowers the binding energy so that the binding can happen at low energy and the reaction can be rapid, and uh, more and more molecules can be treated with the enzyme. Okay. Properties continues, they are very specific towards substrates and products. Enzyme activity can be modulated by non-substrate molecules such as allosteric control, covalent enzyme, and modifications. In few specific cases, enzyme amount can be modulated by synthesis or degradation. Okay. So the properties of enzymes continues. They should be geometrically complementary. Electronic complementary should also be there, and stereospecificities also should be there so that the structure is maintained and enzyme can work in its active form. Okay. Next what we learned in the molecular biology course is about the heredity. So basically the flow of information from parents to their children or from one generation to next generation is what is called as heredity. So heredity is also called as the inheritance. It is the passing on of traits from parental generation to their offsprings, either through asexual reproduction or sexual reproduction. The offspring cells obtain the genetic information of their parents. Okay? They obtain the genetic information of their parents from their parents. Okay. प्रॉपर्टीज क्या क्या हो सकते हैं जेनेटिक मटेरियल की? सो फॉर अ मॉलिक्यूल टू बी कंसिडर्ड एज अ जेनेटिक मटेरियल it must accomplish certain properties. One is stability. DNA is very stable compared to RNA. Heritability and expression, which happens in case of the DNA. Mutation, rare mutations are also there. And replication, it can self replicate. Okay. So DNA has all the properties which are important for a material to be genetic material. So these are the four main properties of that. Okay. Next is the Griffith's experiment to prove that DNA is there is something that is getting transferred from one generation to next generation, so that the offsprings are also carrying the same information as their parents. ठीक है. So Griffith identified two different strains of Streptococcus pneumoniae bacterium, S and R. ठीक है. S strain is the smooth strain which is a virulent and pathogenic strain. Avirulent strain is the R strain, non-pathogenic strain known as R strain. S strain has smooth outer coat made up of the polysaccharides, while the R strain lacks this polysaccharide coat and therefore its surface appears rough. Okay, so basically there in this experiment they have proven that and information travels from one generation to next generation through some molecule which carries this information from one generation to next generation. ठीक है. Next is the thing that we learnt about is the genetic organization in the cells. Each DNA molecule is associated with the proteins, and each DNA molecules and its associated proteins together make what a well arranged chromosome. ठीक है. So chromosome is a complex compactly arranged DNA. ठीक है. That is what the chromosome is, and these are the different stages through which DNA passes so as to make a compactly compactly arranged chromosome. ठीक है. तो ओपन चेन स्ट्रक्चर पहले ये बनाता है, बीडिड अपीयरेंस ये बनाता है, देन सोलेनॉइड ये बनाता है, एंड देन फाइनली इट फॉर्म्स द क्रोमोसोम. ठीक. Next comes what the central dogma of biology कह लो, life कह लो, but it is the central dogma of biology. Sorry. The protein is made up of the amino acids, and every protein has unique amino acids arranged in a specific sequence. The information to synthesize these proteins with unique amino acid sequence is provided by the nucleic acid present within the nucleus, because nucleus contains the DNA and DNA contains all of the information. ठीक है. So in a preset sequence, DNA present in nucleus give rise to a specific RNA sequence, and that in turn guide the cellular machinery to synthesize proteins. ठीक है. So basically what is central dogma of life? It is from DNA RNA is formed, from RNA protein is formed, and DNA can also replicate itself. So this is a central dogma of biology. Information always flows in this direction in the biology. ठीक है. Next comes replication is also what we have learned in the last few lectures. So DNA replication is a biological process that helps to transmit the DNA, a genetic information from from generation to generation by producing two identical DNA strands of daughter cells from daughter stranded parental DNA. ठीक है. इंपॉर्टेंस क्या होगी रेप्लिकेशन की? रेप्लिकेशन इज a way of duplicating all the genetic information from all living parent organisms to daughter organism, thereby helping maintaining the genetic materials intactness and the organism's survival, basically to prevent the mutation. ठीक है. Features of a prokaryotic replication. Now here the replication will also be different in case of both prokaryotes and eukaryotes. So in prokaryotic replication, an origin of replication is there from where the actually replication starts. ठीक है. So we are not again going into the detail of the origin of replication. Next is the DNA replication is the mechanism by which cells duplicate in genetic material, ensuring that each newly formed cell receives an accurate copy of original DNA. This process is essential for growth, development, and transmission of genetic information from one generation to next generation. ठीक है. And DNA replication involves various enzymes and proteins working together to unwind and separate double-stranded DNA molecule. They synthesize new complementary strands and ensure the fidelity of copied genetic information. ठीक है. So again we are not going into the details of the eukaryotic replication. Next comes the transcription. So formation of DNA from DNA is called replication. Formation of RNA from DNA is called as the transcription. ठीक है. So every cell mainly contains three types of RNAs. tRNA, which is a transfer RNA, ribosomal RNA, rRNA, messenger RNA, mRNA. Synthesis of RNA from DNA templates with the help of DNA dependent RNA polymerase is known as transcription. It occurs unidirectionally, in which chain is synthesized 5 prime to 3 prime. The segment which is transcribed from DNA is known as transcriptional unit. Okay, so that will be called as what? Transcriptional unit. In eukaryotes, monocistronic transcription unit occurs, in which coding sequence proteins for only one polypeptide, but in prokaryotes, polycistronic transcription until occurs, in which uh, transcription unit occurs in which coding sequences present for more than one polypeptide. Okay, so alternative splicing happens in case of eukaryotes. That is something which does not happens in case of the prokaryotes. ठीक है. Transcription continues. So it is the first base pair from where transcription starts, and it is called as the start site, and it is generally methionine, AUG. AUG is mostly the start site in case of the DNA, or in case of the DNA. It is a non-template nucleotide at the 5 prime end or minus direction. Sequence before the start point is called as the upstream. Below the start point, it is called as the downstream. Okay? So DNA is a double-stranded structure. During transcription, only one strand is transcribed, so that transcribed sequence is identical with one strand of DNA, known as coding. On the other, complementary strand is known as the template or antisense strand. So coding strand is called as coding strand because whatever strand is formed from replication will be exactly similar to the coding strand. Okay, because both of them are complementary. Next, we learned about operons. What are operons? How they work? So, an operon is a genetic regulatory system mostly seen in prokaryotes and bacteriophages, in which a group of structural genes are transcribed together under the control of a single promoter. So, the promoter here in operons is a single promoter. Okay, generally operons are very common in prokaryotes and bacteriophages, but it is also found in some eukaryotes. So the main difference is that the expression of prokaryotic operons leads to polycistronic mRNA, while eukaryotic operons lead to monocistronic mRNA, similar to the eukaryotic cells. Okay? So this is what is operon is. Next is the translation. So we learned three things in the central dogma of biology: the replication, the formation of RNA from DNA, which is called as transcription, and formation of the whole protein from the RNA is called as the translation. So for translation, there are three RNAs that are involved: rRNA, tRNA, and mRNA, which is the messenger RNA. ठीक है. ट्रांसलेशन करने से पहले you should know what is a genetic code. So mRNA is the random sequence of nucleotides differentiated by the bases attached to them, which are uracil, adenine, cytosine, and guanine. Three nucleotides together code for specific amino acid and are called as the codons. Okay, so codons are actually formed of three nucleotides. Why three nucleotides? Because when the calculations were done, it came out that only a tri nucleotide thing can act as a codon as an information for the proteins. Uh, Genetic code properties here continues. So genetic code is a triplet code called as the codon. Each coding sequence has start and stop codon to initiate and terminate translation respectively. Usually start codon in AUG, which codes for methionine, and stop codon at UAA, UAG, and UGA. Okay. So in some cases, start codons are GUG or UUG. The code is unambiguous, which suggests that code is for only one amino acid. There is no comma or gap in case of the codons. They are written as such and in capitals. Okay. So these were some of the properties. Translation ki mechanism mein, again we are not going into the detail. In eukaryotes there is only one start codon, while in prokaryotes there are many. Eukaryotic cells need more initiation factors than prokaryotes. ठीक है. Next comes the blotting techniques. Then we came to the techniques. What are the different techniques that are used in molecular biology? So for DNA it is southern blotting, for RNA it is northern blotting, for carbohydrate type of post translational modification it is the fast eastern blotting, and western blotting is for protein. ठीक है. So this is for western blotting. So next is the polymerase chain reaction. So to amplify a lot of double-stranded DNA molecules fragments with same identical size and sequence by enzymatic methods are the and the cycling condition. So PCR chain is basically the amplification of whatever your gene of interest is under the situations similar to the cell. ठीक है. So DNA is a complementary in nature. It was proven there in the lecture. We are not going into the details. Again, no details about the DNA replication. It is the initiation, elongation, and termination. Initiation is to prepare DNA for synthesis. Elongation 2 is synthesis phase. Elongation 3 is stopping of DNA synthesis. Now, the formed DNA or formed gene of interest will be released from the in the solution. Okay. So this was the polymerase chain reaction defined here. We are again not going into the details. Next is the real-time polymerase chain reaction. So real-time polymerase chain reaction combines the amplification of target DNA and its decision in single step, that is all collects the data throughout the amplification in itself. ठीक है. Real time PCR molecules that combines the amplification of gene of interest with the intensity of fluorescence just to see the fluorescence. So these kind of many experiments are done, and RT-PCR is the one which will tell you then and there only if your gene has amplified or not. ठीक है. Next is the thing of molecular cloning, how cloning is done. So cloning is basically getting your gene of interest inserted inside some protein producing vector so that you can produce protein also, you can produce antibodies also, and you can also go for different experiments. Okay. Next is the steps in the molecular cloning. So identification of gene will be done, genomic library, cDNA library as screening. Then the cloning will be done. Then the DNA library will help you find that, and the overexpression will tell you what exactly the protein is. Okay, next comes the genetic disorders. So there are many genetic disorders that are still working in the world, and these genetic disorders are basically a single point mutation most of the cases. So let's go in the world of genetic. क्या था हमारा टॉपिक? सॉरी गाइस. जेनेटिक डिसऑर्डर्स. ठीक है. So recombination या core of genetic engineering. Recombination is a genetic process involving the breaking and recombining of DNA fragments, resulting in the creation of a new combination of alleles. This mechanism opens at the gene level, fostering genetic diversity that has been uh, that mirrors variation in DNA sequences among the organisms. ठीक है. Next is the type of recombination reaction that can happen. So it can be homologous, non-homologous. Homologous is bacterial or eukaryotes. Non-homologous has many names. Okay. So drawbacks of traditional method used in genomics is lack of precision, low efficiency, time consuming, limited editing scope, and offer off target effects can also be seen if the gene has not inserted properly. Next, I hope you guys have taken the lectures uploaded by sir. So this is the your work from here starts for week 12 that we'll be discussing tomorrow. So these are the enzymes that are being used in the food industry for different purposes. You guys must have again read it in the video, so I'm not going into the details. But yes, they are being used in the food industry for many purposes. Okay, gene therapy is actually production and supply of the recombinant proteins is in a is a temporary solution of the treatment of a disease condition. In another approach, human expression system is used to produce the proteinaceous factor after inserting the recombinant clone into the human cells or inside the human body. ठीक है? Recombinant DNA is packed into the appropriate DNA delivery system to deliver the gene into the human cells to correct the mutated genes or encode a therapeutic protein drug to provide treatment. ठीक? Next was the gene therapy. It can be of two types, somatic gene or germline gene therapy. Okay, so in somatic gene therapy, in this therapeutic approach, the therapeutic genes is transferred into the somatic cells as per the requirement of the individual to treat the functional defects. This treatment does not move to patient's offspring or next generation. While in case of the germline gene therapy, in this therapeutic approach, germ cells, sperms or egg cells are transformed by the introduction of the required gene to produce the protein or correct the mutated gene. This allows the treatment to move to the patient's offspring or next generation. Although the approach seems promising in providing long term solution to treat genetic disorder, but there are several ethical technical reasons and possible future risks to continue with this kind of gene therapy. So this was all about the course. I hope you guys enjoyed the course. You saw all the videos, you learned many things from the course. And if you have any questions, I'm open for questions. You can ask me the questions. Any questions, anyone? You can ask me the questions. So this was all the course that you guys must have taken while doing the classes. And all the very best for your exam. When is your exam? I haven't checked the date yet. हाँ जी, किसी को कोई सवाल नहीं पूछना? सबको सब कुछ समझ आया? किसी को कोई कंफ्यूजन हो, कोई टॉपिक समझ नहीं आया हो? वैसे तो हम कल भी मिलेंगे, पर आज पूछना है तो आप लोग आज भी पूछ सकते हैं. Anyone, any question? I'm waiting for your queries, everyone. कोई तो कुछ पूछो, नहीं पूछना? सबको सब समझ आया है? ठीक है, सबको सब समझ आया तो अपने एग्जाम में अच्छे से लिखना. ठीक है, वी आर देन फिनिशिंग दिस सेशन. और आपको कोई भी सवाल हो, कुछ रह गया हो, फिर भी पूछना. तो कल की क्लास भी होगी सेम टाइम 6 o'clock. यू कैन आस्क मी द क्वेश्चंस देन. ठीक है, now you guys can leave the meeting if you guys don't have any questions. Thank you so much for joining. Now you can leave the meeting. ठीक है। Okay, guys. Bye bye. Thank you so much for joining.